Hello, students. So, we will start with NCRT exercise of chapter 13, that is plant growth and development. So, this is a disclaimer. So, this will be our learning objectives. We will explore and understand the fundamental concepts of chapter 13, that is plant growth and development, by working through the NCRT exercise. This will be our index. So, we will solve question number 1 to question number 8 of NCRT exercise for this chapter. Let's start with the questions. So this is question number 1. Question is define growth, differentiation, development, de-differentiation, redifferentiation, determinate growth, meristem and growth rate. So this question will cover NCRT section 13.1 and 13.2. Let's start with the answer. So talking about growth, it is an irreversible and permanent process. It is never changing process. It is a permanent. It is a permanent process. A process that cannot be changed. It is accomplished by an increase in the size of an organ or organ parts or an individual cell. So if we see an organ on any kind of cell or any organ part is increasing in size, then we can say that particular organ or cell or organ part is growing, and that's a part of growth. So it is main important points here are it is an irreversible permanent change, and second one is increase in the size. We if something is growing, then we will see that the size is increasing. So these two things you have to keep in your mind. Let's talk about differentiation. During this differentiation, these cells undergo structural changes in their cell walls and protoplasm. Okay, think of it as remodeling a room to serve a new purpose. Maybe turning into a bedroom, maybe turning a bedroom into a study room. Okay, so the cell changes its structure to do a new job. As a result of differentiation, cells become mature and are able to perform specific functions. For example, some become xylem cells to transport water, while other become leaf cells to carry out photosynthesis. Each cell can find its own carrier path in the plant. So it's a process in which cells derived from apical meristem, typically root and shoot meristem. So apical meristem further are of two types: root meristem. If the apex if it is present in the apex of the root, then it will be root meristem, and if it is present in the apex of shoot, then it will be shoot meristem. Okay, so apical meristem and the cambium. So it can undergo structural changes in the cell wall and the protoplasm. Cells here become mature to perform specific functions. You have to keep in your mind that here cells will not divide. They have some specialization, they got some designation, they got some specialization, and they will do that particular or that specific job. So here they cannot divide. They get mature, and here and therefore they perform specific functions. So here cells got specified. Let's continue the solution. Talking about development, it refers to all the changes that occur in an organism during its life cycle. It's not just about getting bigger, it's about the transformation from one stage to another, like from a seed to seedling, then to a mature plant. So it refers to the various changes occurring in an organism during its life cycle. For example, when a plant increases in size, that's a part of development. But remember, development also includes changes in shape, structure, function, not just size. So you have to keep in your mind that development also includes other aspects like changes in shape, structure, function. Okay, we are not specifying any just size. Okay, here. Though these changes range from the germination of seeds all the way to senescence, which is the aging and eventual death of the plant. Each stage is essential for the plant survival and reproduction. So take a look at this figure. It shows the sequence of developmental process in plants. Notice how the cell starts simple and becomes more specialized over time. So this is the sequence of the development process in plant. It during the development it undergo differentiation, de-differentiation, and redifferentiation. Okay, we will study them one by one. First, development. Talking about development in broad aspect, it refers to the various changes occurring in an organism during its life cycle. If you talk about if you talk about example, then increase in the size, shape of a plant is an example of development. Here, change ranges from the germination of seeds to senescence. Let's continue the answer. Okay, talking about de-differentiation. De-differentiation is when when living differentiated cells have lost the ability to divide, can regain it. So here in differentiated cell, what we have learned that they lost the ability to divide. Okay, but here those differentiated cell who once lost the ability to divide can regain this ability to divide. Okay, so this is like a specialized worker being retrained to a different job. So it's a process in which living differentiated cells that have by now lost ability to divide regain the power to divide under certain conditions. Okay, they regain the power of division. That and in differentiation, what happened, they lost the ability to differentiate. They lost the ability to divide. So this is the sequence that is showing the stages of developmental process that we talked about. Talking about redifferentiation, it's when redifferentiated cell lose their capacity to divide, they become specialized again, taking on new roles in the plant. So this is redifferentiation. Like, it's a process in which de-differentiated cells who lose their ability to divide, here cells become mature, and here cells gain the here cell got specialized to do a new job. Okay, they lost the ability to divide, and they got specialized to do a new job. Okay, now talking about determinate growth. Determinate, the word itself says is saying that fixed or limited. So determinate growth means that the growth is limited. Once the structure reaches its final size, it doesn't grow any further. For example, animal and plant leaves stop growing after reaching maturity. This is different from roots or stem, which can keep growing indefinitely in some plants. So this is the sequence of the developmental process in the plant cell. Talking about meristem, in plants, growth is restricted to specialized regions where cell divisions takes place. These regions are the meristem, and they are like the plant's growth engines. So in plants, growth is restricted to specialized regions where active cell divisions takes place. Here, cells continuously divide. Such a region is called as meristem. Remember, without meristems, plant wouldn't be able to grow new tissues or repair damage. So there are three types of meristem: apical meristem, lateral meristem, and intercalary meristem. So apical means apex. It is related to apex. It is present in the apex. Lateral is important for width, for increasing the width. And talking about intercalary, it is important for elongation. Okay, it is present in between. Let's talk about other terms. Growth rate. So growth rate can be defined as the increase growth in plants per unit time. In other words, it's how much a plant grows in a given period, like centimeter or millimeter per day. The growth rate can show an increase that may be arithmetic or geometric. Okay, arithmetic means a steady regular increase, while geometric means the increase gets faster and faster, like compound interest in a bank account. So a sigmoid curve is a characteristic of living organism when growing in a natural habitat. Now this is second question, and the question is, Why is not any one parameter good enough to demonstrate growth throughout the life of a flowering plant? So this question is covering NCRT section 13.1.2. Let's see the answer. In plants, growth is said to have taken place when the amount of protoplasm increases. Protoplasm, what is protoplasm? It is the living content of a cell. So, an increase here means the plant is truly growing at a cellular level. Now, measuring the growth of a protoplasm involves many parameters. It's not just about one thing. Scientists need to look at several aspects to get an accurate measurement. Let's list out some of these measurements. First, we have the weight of the fresh tissue sample. This tells us about how much living sample or how much living material and water is present in a plant cell at the given period of time. Next, the weight of the dry tissue sample is measured. By removing all the water, we can see how much solid material the plant has produced, which is more stable indicator of the growth. We also look at different differences in length, area, volume, and cell number during the growth period. These measurements help us understand how the plant is expanding and developing over time. So, measuring the growth of plants using one parameter doesn't provide enough information. Each parameter tells us something unique, and relying on just one parameter can be misleading. Hence, it is insufficient for demonstrating growth. To truly understand how a plant is growing, we need to consider all these different parameters together. Okay. So, parameters like weight of the fresh tissue sample or weight of the dry tissue sample, they cannot be accurate. They can show us different results. So, we cannot rely on a particular parameter. We have to consider all parameter together to demonstrate or to measure growth throughout the life of the flowering plant. So I have to consider all of the parameters. We have to consider most of the parameters, because one specific parameter cannot give us accurate results. So this is question number 3, and the question is, describe briefly arithmetic growth, geometric growth, sigmoid growth curve, and absolute and relative growth rates. So this question is covering NCRT section 13.1.4. Let's see the answer one by one. Let's focus specifically on arithmetic growth. This is the first type of growth mentioned in the question, and it's important to understand its basic features. In arithmetic growth, only one of the daughter cells produced during cell division, and during cell division continues to divide. So you have to keep in your mind that during this, only one daughter cell, others are not involved here. Only one daughter cell only one of the only one of the two daughter cells produced during cell division continues to divide. Only one daughter cell here divide. Okay, and the other cell stops dividing and start to mature. So this is like a relay race where only one runner keeps going while the other finishes their part. So in arithmetic growth, in broad aspect, we can say one of the daughter cells continues to divide. So that cell that doesn't keep dividing begins to differentiate, meaning it takes on specialized function in the plant. This is similar to how in a company some employees move to new projects while others settle into their roles. So you have to keep in your mind that here only one daughter cell is dividing and the other got specialized to perform different job, to perform different function. A classic example of arithmetic growth is the elongation of roots at a constant rate. Imagine a root growing steadily like a clock ticking at a regular pace, and this steady growth is what defines arithmetic growth. So the elongation of roots at a constant rate is an example of arithmetic growth. So this is the picture that is showing arithmetic growth. This is showing how one of the two daughter cell is dividing and the other cell got specialized to do other job. Talking about geometric growth, this is the second type of growth pattern mentioned in the question, and we have to look into the functions of the or features of this growth pattern. So it starts with the slow phase. In the beginning, the plants increase in size is gradual, almost like a gentle slope on a hill. So we have to characterize this type of growth by a slow growth in the initial stages, and a rapid exponential growth during the later stages. So this happens because the daughter cells produced during mitosis keep dividing. So the daughter cells derived from mitosis retain the ability to divide. The growth slows down because of limited nutrient availability, because any everything or resources is limited. So due to limited resources, after exponential period, after exponential growth, the growth slows down, okay, due to depletion of the resources. So this is the picture that is showing geometric growth. It is, this growth is characterized by the slow increase. First, the increase is very slow, then it increases exponentially, and after reaching certain level, or after reaching certain exponential rate, it it get limited to certain amount. Why? Due to depletion of resources, due to less availability of resources. So this is the pattern, this is the picture that is showing the geometric growth pattern. So let's talk about sigmoid growth curve. The sigmoid growth curve describes how living organisms like plants grow in their natural habitat. The S shape shows the growth starts slowly, speeds up, and then slows down again as the plants matures. So the growth of living organisms in their natural habitat is characterized by S-shaped curve, which we commonly call as sigmoid growth curve. So let's look at three main phases of sigmoid curve. First one is lag phase. So this is when the plant is just starting to grow, adjusting to its environment and preparing for rapid growth. Next is the exponential phase, also called as the phase of rapid growth. Here the plant grows quickly, making the most of the available resources. Finally, we have the stationary growth or stationary phase. In this phase, the plant's growth slows down and eventually stops, usually because resources become limited or the plant reaches maturity. So this S curve is characterized by three phases. For the three phases, lag phase, which is a slow phase, log phase, or exponential phase of rapid growth. Here, the growth is here growth is increasing exponentially due to ample of resources. Okay, and third one is stationary phase, due to limited resources, due to depletion of resources, due to less resources, or due to maturity, the growth become limited, and so it reaches stationary phase, or stable bio or stable growth. Let's see the other part. Exponential growth can be exponential growth. Talking about exponential growth, so it is a key feature of geometric growth, and it's important to understand how we can express it mathematically. So this is the mathematical formula, or how we can express this mathematically is W1 equals to W naught e raised to the power ERT. So this equation help us to calculate the final size of a plant organ after a certain period given the initial size and growth rate. So let's break down what each part of the formula means. So here W1 stands for final size, W naught is the initial size, R is the growth rate, T is the time of growth, and E is the base of natural logarithms. Each part here plays an important role in determining how quickly and how much a plant grows. So let's focus on absolute and relative growth rates. Absolute, talking about absolute growth rate refers to the measurement and comparison of total growth per unit time. So For if you talk about example, if a plant grows 3 centimeter in 3 days, its absolute growth rate is 1 centimeter per day. So it refers to measurement and comparison of total growth rate per unit time. Relative growth rate refers to the growth of particular system per unit time, which is expressed on a common basis. So this means we look at how each the plant grows compared to its initial size, which helps us compare growth between different plants or organs. For example, if we say per unit initial parameter, we are talking about relative growth rate. If a small seedling doubles in size, that's a high relative growth rate, even if the actual increase is small. So here we are visualizing two leaves A and B, which are different in size. This helps us to compare how growth can look different depending on the starting size of the organ, even the increase is the same. So both leaves A and B show the same absolute increase in the area, which is 5 square centimeters. So this means that if you just look at the total area added, both leaves grow by the same amount regardless of their starting size. However, when we look at relative growth rate, one leaf shows much higher value. This is because relative growth rate compares the increase to the original size, not just the amount added. So for leaf A, if we talk about leaf A, we started at 5 square centimeters and grow by 5 more. The relative growth rate is 5 divided by 5 means 100%, which equals 100%. This means leaf A double in size, which is a huge change compared to its starting point. So for if we talk about leaf B, which started at 50 square centimeters and also grow by 5, the relative growth rate is 5 divided by 50 times 100%, which equals to 10%. So even though both leaves grow by the same amount, the impact is much less for the larger leaf. So here we see that leaf A has much higher relative rate, that is 100%, compared to leaf B, which is only 10%. So this highlights how relative growth rate gives us better sense of how much a plant part is growing compared to its original size, not just the total increase. So you have to keep in your mind the difference between absolute and relative growth rates. So this is question number 4, and the question is, list 5 main groups of natural plant growth regulators. Write a note on discovery, physiological functions, and agricultural or horticultural applications of any of them. So this question will cover your NCRT section 13.4. So just like us, we have hormones in our body for chemical control and coordination. Same like us, plants do have hormones. We call them plant growth regulators. So we have to write a note on their discovery, how they discovered their physiological aspects, and their horticultural or agricultural applications. So let's learn them one by one. So let's begin our solution by defining plant growth regulators. So these are chemical molecules produced by plant themselves, and they have big impact on how the plant functions. Okay. So there are talking about their numbers. So there are 5 main plant regulators that are. So first is auxin. Auxin is crucial for cell elongation, root initiation, directional growth. So they are the first plant hormone discovered, and they help plants bend toward light, a process called photoperiodism. Then we have gibberellin. These hormones are responsible for stem elongation, seed germination, and flowering purpose. Gibberellin can make plants grow taller and are used in agriculture to increase fruit size. The third group is cytokinin. These promote cell division and are important for shoot formation and delaying leaf aging. So cytokinin are like the youth serum for plants, keeping tissues young and active. So to keep them in your mind, they keep the tissues or cells young. Fourth is ethylene. Ethylene is just opposite to Cytokinin. Cytokinin keeps the leaves or organ young, but it is involved in ripening. Okay, so it is a gaseous hormone that controls fruit ripening and leaf abscission. Okay, so if you've ever seen banana ripen quickly when kept together, that's ethylene at work. Due to its gaseous state, it is more impactful. Now, finally, we have abscisic acid. This hormone helps plants cope up with stress like drought and induces seed dormancy. It's sometimes called as stress hormone of plants. So, let's continue with the auxin. Auxin are crucial for plant growth and development. So, let's look at the discovery. So, Charles Darwin and his son Francis were the first to notice the effects of auxin. Their experiments with canary grass coleoptiles laid the foundation for our understanding of, of the plant regulators or plant hormones. So, they observed that the coleoptiles of canary grass would bend toward a light source. This bending is visible response that plants make to optimize their exposure to sunlight. After several experiments, they concluded that some substance were produced at the tip of the coleoptile. So, this substance was responsible for signaling the plant to bend toward the light. This substance produced at the tip was found to responsible for bending movement. It's like the tip is the commander center, sending structure instruction to the rest of the plant. Eventually, scientists managed to extract this substance from the tip of the oat seedlings. They named it auxin, and this marked the beginning of our understanding of plant growth regulators. So this is the picture which is demonstrating the tip of the coleoptile, which is the source of oxygen. So here arrows indicate the direction of light. Let's talk about physiological functions. First, auxin control plant growth. They help cells elongate, which is essential for the plant to grow taller and for roots to extend deeper into the cell. Next, auxin causes the phenomenon of apical dominance. This means the main shoot grows more vigorously than the side branches, helping the plant reach more sunlight. Just like a tree growing taller in forest to outcompete its neighbors. Auxin also controls cell division in vascular cambium and xylem differentiation. This is like building new highways in the plant for water and nutrients to travel efficiently. Additionally, auxin induce parthenocarpy, which is development of fruit without seeds, and they prevent abscission or the dropping of leaves and fruits. So this helps plant retain their fruits and leaves longer, which is especially useful in agriculture. Here you can see an image illustrating apical dominance. Notice how the main shoot is dominant and the side branches are suppressed. This visual helps us understand how auxin direct plant growth. Let's see the horticulture applications of auxin. First, auxin are used as rooting hormones in stem settings. When gardeners want to grow new plants from cuttings, apply auxin encourages the cuttings to develop roots more quickly and successfully. It's like giving the plant to boost. It's like giving a plant a boost to start its own life. Another important use is weedicide. So the synthetic auxin 2,4-D is used to kill broadleaf dicot weeds without harming crops like wheat or rice. This selective action helps farmers manage weeds very efficiently. So it can also induce parthenocarpy in tomatoes. This means that they can produce seedless tomatoes, which are often preferred in the market, and it's a way to get fruit without the need for pollination or fertilization. So it induces parthenocarpy. Auxin are also used to promote flowering in crops like pineapple and lychee. By applying auxin at the right time, farmers can encourage these plants to flower more uniformly, which helps with harvesting and improves the yielding. Let's continue the solution. Talking about gibberellic acid, Japanese farmers noticed a disease called Bacon or foolish seedling disease. So this was the first clue that something unusual was happening in their rice fields. In this disease, rice seedlings grow much taller than the normal plants, but they were also thin and pale green. It's like the plants were growing too fast for their own good. So after so many experiments, scientists found that this condition was caused by a by a fungi, which we call as Gibberella fujikuroi. So this fungus was infecting the rice plants and making them grow abnormally. Eventually, researchers isolated the active substances from the fungus and identified it as gibberellic acid. So this discovery helps us understand how plant hormones can dramatically affect growth. So let's focus on physiological aspects or physiological functions of gibberellin. First, gibberellin can cause elongation of internode. This means that they can help the stem grow longer between the points where leaves are attached, which is called as node. Think of it as stretching out plants' spine so it can reach more sunlight. Gibberellin promote bolting in rosette plants. This is very important. Bolting. What is bolting? Bolting is when plant suddenly grows a tall flowering stalk, which is important for reproduction. So this is especially noticeable in plants like lettuce or onions. It also help induce seed germination by breaking seed dormancy. So they trigger the synthesis of hydrolase enzyme, which digests stored food in the seed, giving the young plant the energy it needs to grow to start growing. So it's like unlocking the pantry so the seedling can have breakfast and starts its day. So let's look at the horticultural applications of gibberellin. It is used to increase the sugar content in sugarcane by making the internodes longer. The plant stores more. This way, the plant stores more sugar, which is crucial for high yields in sugarcane farming. So, this is a great examples of how understanding plant hormones can really impact food production. It also increase the length of grape stalk. This makes grape bunches less compact and improving airflow and reducing the risk of fungal diseases. So it's a simple change that leads to healthier and better quality grapes. Another application is improving the shape of apples by using gibberellin. Uh, it can produce apples with more uniform and appealing shape. So which is very important for sales or marketing. So this shows how plant hormone can influence not just growth but also the appearance of the fruits. By using different regulators, we can also change the appearance of the fruit and make it more appealing. It delays senescence, which means they slow down the aging process in leaves and fruits. This keeps the plant productive for longer and helps maintain fruit quality during storage and transport. So it can also hasten maturity and induce seed production in juvenile conifers. This is especially useful for in forestry, where faster seed production can help with replanting and conservation efforts. So it shows how plant hormones are valuable just beyond food crops. Let's talk about cytokinin. Let's start with the discovery. So F. Cook and his team noticed that when they added certain substances to tobacco callus tissue, the cell began to differentiate. This was the key clue that something in those substances was promoting growth. So they experimented with extracts from vascular tissues, yeast, coconut milk, and even DNA. When these were added to the culture medium, the callus differentiated, showing that the growth promoting factor was present. This led to identification and discovery of cytokinin. It was a major step forward in understanding how plant cell control cell division and development. Talking about physiological aspects, they primarily promote the growth of lateral branches by inhibiting the apical dominance. So it acts opposite to auxin. Auxin leads to apical dominance and it inhibit the growth of lateral branches, whereas this is opposite to auxin. It is inhibiting apical dominance but it leads to the growth of lateral branches. They also helps in the production of new leaves, chloroplasts, and adventitious shoots. They help in delaying senescence by promoting nutrient mobilization. They do have important role in senescence. They delay senescence. And how they do it? By promoting nutrient mobilization, by improving nutrient mobilization. Let's talk about horticultural applications. So they are used for preventing apical dominance because they promote lateral growth of lateral branches. They are used for delaying senescence in leaf. Let's continue the answer. Let's talk about ethylene and how it was discovered. People noticed that unripe bananas ripened faster when stored with ripe bananas. This everyday observation leads researchers to wonder what was causing that effect. Eventually, scientists identified that the substance responsible for promoting ripening was ethylene. So, this discovery helped us understand how fruits communicate and coordinate ripening. Let's talk about physiological functions of ethylene. So it helps in breaking seed and bud dormancy. This means it signals seeds and buds to start growing after a period of rest, much like an alarm clock waking you up in the morning. So it promotes rapid internode elongation in deep water rice plants. This allows rice plants to keep their leaves above water during floods, almost like a plant stretching to keep its head above the water. So it promotes rapid internode elongation, and this leads to increasing the length of the plant or rice plant. It promotes if you talk about another main function, it is the thylene promotes root growth and the formation of root hairs. So this helps the plant absorb more water and nutrients from the soil, uh, similar to how move fingers help you grab things better. So it promotes root growth and formation of root hairs. Ethylene also promotes senescence and abscission of leaves and flowers. Senescence, what is senescence? Senescence is aging process, and abscission is the dropping of leaves or flowers. So it acts it acts as like a signal for the plant to shed old parts. It hastens the respiration rate in fruits and enhances fruit ripening. This is why banana and tomatoes ripen faster when they keep together, because they ripen. Fruits contain ethylene, and due to its gaseous state, the impact is also great. So you can see the image how ethylene enhances ripening. Notice how the fruit change color, texture as they ripen, all thanks to our ethylene. So talking about horticultural application, it is used to initiate flowering and synchronize fruit set in pineapples. This means farmer can get all the fruits to develop at the same time, making harvesting easier and more efficient. It also induces flowering in mango. By applying ethylene, growth can control when mango trees flower, which helps in planning the harvest and meeting market demand. Ethophone, a chemical that releases ethylene, is used to ripen fruits like tomatoes and apples. So this allows fruits to be picked before they are fully ripe, reducing the damage during transport and ensuring they reach consumers in perfect state. It also accelerates the abscission or dropping of flowers and leaves in crops like cotton, cherry, or walnut. This is helpful for managing harvest and maintaining plant health. It also promotes the number of female flowers in cucumbers. More female flowers means more fruits, which increases the yield for farmers. So we will continue this solution. Let's talk about abscisic acid. So, in the talking about discovery in the mid 1960s, three different researchers discovered substances called inhibitor B, abscission 2, and dormin. Each of these were found in different plants and seem to have similar effects like stopping growth or causing leaves to fall. Later on, scientists realized that these substances were actually chemically similar. They were all forms of the same compound, which are then named as abscisic acid or ABA. Cornforth and his team in 1965 showed that that domain and abscissin 2 were actually the same chemical. So this discovery unified the understanding of these growth inhibitors under one name. Because these substances were linked to the process of abscission or dropping of leaves and fruits, the name abscisic acid was chosen. So, this name reflects its main observed effect in plants. So, talking about its physical features or aspects, it acts as an inhibitor to plant metabolism. It also stimulates stomatal closure during water stress. That's why it is also called as stress hormone. It induces seed dormancy. It also induces abscission of leaves, fruits, and flowers. So, first, AB is known to inhibit seed germination, and this property is useful for extending the shelf life of seeds as it prevents them to it prevents them from sprouting too early during storage. So, this is the first horticultural application of abscisic acid, that it is known to inhibit seed germination, which can be useful for understanding the shelf life of the seeds. It also plays a complex role in fruit ripening. It affects the color, texture, flavor, and aroma of fruits, which is important for both farmers and consumers who want quality fruits. Another application is that AB can be used to regulate plant growth. For example, it can promote root growth and inhibit excessive vegetative growth, help growers to manage the size and health of their plants. It can also promote root development in cuttings. This is especially helpful in vegetative propagation, making it easier to grow new plants from cuttings in horticulture. So this brings us to the end of this question. So this is question number 5, and the question is why abscisic acid is also called as stress hormone. So this question is covering NCERT section 13.4.3.5. Let's see the answer. So it is called as stress hormone because it triggers various responses in plants when they face stress. So when they face stress, they induces various responses with respect to the surrounding stress. Building on that, abscisic acid doesn't just trigger a response; it actually increases the plant ability to tolerate stress. It's like giving the plant a set of tools to handle difficult conditions, making it stronger and more resilient. A specific example is how abscisic acid causes the stomata to close during water stress. Remember, stomata are tiny pores on the leaves that let water and gases move in and out. So, by closing these, the plant conserves water just like we close windows to keep heat or cool air inside our homes. So it also ensures that seeds only germinate when conditions are right. It acts like a safety lock, preventing the seeds from sprouting during stressful times and only allowing them to grow when the environment is favorable. Another important aspect is helping seeds survive dry conditions. Abscisic acid induces dormancy, which is like putting the seed into putting the seed into a protective sleeve, so it doesn't lose too much water and can wait for better conditions. So at the end of the growing season, abscisic acid helps the plant prepare for winter or dry periods by inducing dormancies in various parts of the plants. This is similar to how animals hibernate during harsh season or during their hibernation period. So finally, abscisic acid promotes abscission, which means it helps the plant shed leaves, fruits, and flowers. This means the it allows the plant to conserve resources and survive until conditions improve. It's much like a tree dropping its leaf in autumn. So this is question number 6, and the question is both growth and differentiation in higher plants are open. Comment. And this question is covering our NCRT section 13.1 and 13.2. Let's see the answer. Growth and development in higher plant is refers to as open. This means that the plants have the ability to keep growing and developing new parts throughout the lives, unlike most animals. So emphasize that this is a defining feature of plant biology. The reason for this openness is the presence of various meristems. These are the regions in the plants where cells are constantly dividing, much like a factory that never shuts down. Highlight that this continuous cell division is what allows plant to keep growing. Importantly, these meristems are present at different locations in the plant body. So, if you talk about example, you can find them at the tips of roots and shoots, and even in the stem. So, this widespread distribution means that the growth can happen in many places at once, allowing the plant to adapt and respond to its environment. So this is question number 7, and the question is, which one of the plant growth regulator would you use if you are asked to? So we have given different conditions, and we have to know, we have to answer like in these conditions what plant growth regulator we will be using. Okay, so this particular question is covering NCRT section 13.4.3. Let's look into the answer one by one. Let's start with A. Induce rooting in a twig. So to induce rooting in a twig, we use auxin. Auxin, it's like a plant's rooting hormone. They help. They mainly help in new roots grow from the cuttings. So this is why gardeners often tip cuttings in often tip cut in rooting powder contain auxin. For a quickly ripening a fruit, the answer is ethylene. So ethylene is a gas it's a gaseous hormone that acts as a ripening signal, turning green bananas yellow and softening other fruits. So it is majorly responsible for ripening, and it is impactful, it is highly impactful because unlike other hormones which are present in solid or liquid form, it is present in gaseous form, and this form make it more impactful. Okay, so it's used in fruit storage and transport to control when fruits are ripened. To delay leaf senescence, cytokinins are used. I have told you already, we have already discussed that it keeps cytokinin keep these plants young. So it basically keeps leaves green and healthy for longer time, almost like an anti-aging treatment for plants. So this is why florists sometimes use cytokinin to keep cut flowers fresh. To induce growth in auxiliary birds, cytokinins are again the answer. They encourage the dormant birds to grow, leading to bushier plants. This is why pruning, which reduce oxygen levels, can also stimulate bud growth. To bolt a rosette, I've told you already, bolting means gibberellic acid. So you have to keep in your mind bolting equals to gibberellic acid. So gibberellic acid is used here. It triggers the plant to grow tall and flower rapidly, which is important in crops like lettuce or cabbage when we want them to flower for seed production. To induce immediate stomatal closure in leaves, abscisic acid is the key hormone. We have already talked about it, why it is called as stress hormone. So it acts like an emergency signal, telling the plant to close its stomata closely, to quickly very quickly, to conserve water during stress period or during drought. Let's move to the next question, and the question is, what would be expected what would be expected to happen if. So we have given all this is like the previous questions. We have given four different cases, and we have to answer in these cases what we will done supposedly. So this question is covering NCRT section 13.4.3. Let's see the answer one by one. So, like, look at the first part of the solution. What happens when GA3 is applied to GA3 is applied to seed seedlings? So, when GA3 is applied, the seed seedlings show a clear increase in the length of their internodes. Imagine the stem segments stretching out, making the plant look much taller than normal. This leads to an overall increase in the height of the rice plants. It's similar to how some children have growth spurts and suddenly get taller in short period. So let's next consider what happens if dividing cells in a plant stop differentiating. This is a crucial step in plant development. So if differentiation doesn't occur, the plant won't be able to form specialized organs like leaves or stems. It's like having a group of workers who never take on specific job, so nothing gets built. So instead, what forms is a callus, which is just a mass of undifferentiated cells. In labs, scientists use callus tissue as a starting point to regenerate whole plants under the right conditions. We'll continue the answer. So what happens if rotten fruit gets mixed up with unripe fruits? We have already talked about ethylene. The ethylene gas produced by the rotten fruit acts as a signal to unripe fruits, causing them to ripen faster. This is why one spoiled fruit can make the others go bad quickly. Let's look at the scenario. So what happens if you forget to add cytokinin to the culture medium in a plant tissue culture experiment? Without cytokinin you won't be able, C for cytokinin, C for cell division, so you won't be able to see cell division, growth, or cell differentiation in the culture medium. So it's like trying to grow a plant without giving it the signals it needs to multiply or develop. So so thank you, this is the end of the of the session. I hope you have understood the concepts of this chapter through the ncrt exercise questions. So thank you.